Okay, my name is Ian Baldwin, and I'm a director of the Planck Institute for Chemical Ecology in Jena. And I'm going to give you a three-part talk on studying plant ecological interactions in the genomics era. The first part, this part one, is going to be a short biased history of this particular interdisciplinary field. And it's biased because I have the main objective of introducing how the history of the field influenced the development of the particular institute that I'm involved in, this institute in Jena. And I also want to introduce the plant that I am showing right here in this picture, which is Nicotiana attenuata. So the first talk, this one is the history to the field. The second part is going to be about how the plant has responded in a remarkable way when herbivores break through its major resistance trait, namely nicotine production. And the third part is about how the plant deals with its seeds, its sex, and its microbes. This first part is going to have a number of subsections as well. I'm trying to develop in this part an organismic perspective on plants. I'm going to give you a brief history of the field, and I'm going to develop this process that we use in our institute of asking the ecosystem for the function of plant genes and give you an example of an interdisciplinary analysis of plant-pollinator interaction. So that's the outline of where we're going to go today in this particular part one of this talk. So let me first with an organismic perspective on plants. Plants are autotrophic. That means they eat sunlight to fix carbon and get the nutrients that they need. They're also immobile. They have root systems, and mostly they're rooted in the soil and can't run around. So they have to deal with things in the place that they happen to be growing. And the third thing is that they are at the base of the food chain. Everybody eats them. And of course, the last thing a plant really wants is to be eaten, unless, of course, it's at a very particular time, at a very particular stage, when it wants to be eaten so that it can gain the dispersal services of the thing that's eating it, like with things that eat fruit and poop out the seeds in nice places for them to grow. So if you take a course in plant physiology and you study plants in a botany department, you learn that plants are different. They have all sorts of traits that make them different from animals. They can photosynthesize. They have lots of mouths, all these stomata over the surface of their leaves that allow them to take in CO2 and fix it. Their cells are confined in wooden little boxes. They have many meristems throughout, both above ground and below ground, and as a consequence, their decision process is distributed throughout the organism. And they have very complicated life cycles with lots of complicated jargon that goes along with it. And generally, physiologists have thought about plants as growth machines. And that's an interesting thing because it primarily focuses on the process of their autotrophic lifestyle. Now, what I want to focus on in this talk is really another unique aspect of plants, and namely their metabolic exuberance. They make a lot of different types of metabolites. If you take a human being like myself, pop me into a blender, hit mix, and then extract all the compounds you get out of me, you would only find about 3,000 metabolites. But if you do that with any plant you find outside, you will get about 2 orders of magnitude more metabolites in them. They are superb organic chemists. Now, the point I'd like to make is that while these are growth machines, they are able to eat sunlight, fix carbon dioxide, use that carbon dioxide to make metabolites that are important for growth, reproduction, and storage, just like all other organisms. They do another thing with that biosynthetic prowess that they have, and that is they make what is referred to as secondary metabolism. They make all sorts of different compounds that are appearing here, compounds that really don't have anything to do with the normal processes of life, namely that of growth, reproduction, and storage. These are compounds that pretty much were the main problems that synthetic organic chemists tried to solve the structures of and find biosynthetic routes toward, and have been very important in lots of aspects of human life, and particularly in terms of creation of drugs for various medicinal purposes. But of course, plants aren't producing these compounds for medicinal purposes for human beings. They're producing these compounds to solve ecological problems. Now, this idea that plants produce all these different secondary metabolites to solve ecological problems is actually fairly new. If you go back to the time when organic chemistry was beginning to develop its fundamental rules and learning the basic principles of how things work, and namely before World War I and through World War II, up into the 60s, these products would have been thought, if you get to the literature, to be functionally inert. They've been considered sort of byproducts of metabolism, excretion products, flotsam and jetsam on the metabolic beach. And they were even thought to be so neutral, in other words, not have any particular role in the organism's life, that they could be used as chemotaxonomic traits. In other words, ways of figuring out their evolutionary history because they haven't been selected for any particular adaptive purpose. Just like you can use DNA these days to be able to develop phylogenetic trees. These metabolites were used by systematists to understand the phylogenetic and evolutionary relationships among plants because they were thought to be adaptively neutral. Waste products. Well, that all changed in 1959, when Gottfried Frankl from the University of Illinois published this important paper in Science called the raison d'etre for secondary metabolites. And what he basically said is, hey guys, it's not metabolic junk. These are the things that plants use to solve ecological problems. And this idea, published in 1958, generated an absolute enthusiastic outburst of theory from ecologists and zoologists. Now, what ecologists and zoologists did was they developed an idea of plants not as growth machines, but really as Darwinian creatures. Creatures that maximize their Darwinian fitness, and maximizing Darwinian fitness means producing successful grandchildren. So this new perspective on plants as Darwinian creatures, that was basically introduced to the field in 1959 by Gottfried Frankl's paper, generated a profusion of theory generated from ecologists and zoologists. This theory was extremely important in organizing people's thoughts about the sort of functional role that plants, these secondary metabolites play for plants. But the theory had a bit of a problem, and a problem that we've articulated in a paper that I co-authored with Mary Schumann in the Annual Reviews of Entomology, that the theories were frequently posed at different levels of analysis. So along the x-axis, you can see that hypotheses are normally posed either at the ultimate level of analysis, evolutionary origins or functional consequences, or at the mechanistic level of analysis, ontogenic processes and physiological mechanisms. And as you can see on the y-axis, where the different theories are laid out in time as they came, that they were frequently posed at different levels of analysis, and therefore the hypotheses were not posed at the same and didn't really represent significant advances, particularly from the perspective of plant physiologists working in the field, who largely have ignored this large theoretical basis. So, as Paul Sherman has pointed out in an important paper in Animal Behavior, these levels are important to keep straight, and alternative hypotheses must be posed at the same for science to advance itself. Now, in this field, which mixes and spans questions that move from questions about how things work to why things work, it's particularly important to keep the levels of analysis separate, but also that it's particularly important to understand that mechanism informs functional levels analysis. In other words, if you understand how things work, you get the tools that allow you to manipulate the expression of that particular trait and thereby understand its functional consequences, namely what it does for Darwinian fitness. Manipulation is the gold standard for the analysis of function. So this interplay between mechanism and function has been extremely important in this field and is a topic that I will bring up not only in this talk, but in the later two talks as well. Now, it turns out that Gottfried Frankl in 1958 was not the first person to argue that these metabolites were playing a functional role. 100 years earlier, in a little town in Germany called Jena, Germany, there was a researcher named Ernst Stahl who had been working on snails and what determined the ability of snails and slugs to eat plants. And he was basically the first experimental chemical ecologist. He worked on plants that produced various sorts of resistance traits like these little rafide spikes that are inside of the cells of water lettuce, and was able to show that if you treated these leaves with hydrochloric acid, a dilute solution of hydrochloric acid that dissolved those rafide needles, slugs love to eat the plants. And it was really the first experimental demonstration of a resistance trait of a plant, done 100 years before Gottfried Frankl articulated the importance of these metabolites for the ecological function. Now, I'm not the one that discovered this. This was all discovered by Thomas Hartmann in a beautiful paper in PNAS in 2008 and presented in a talk in our institute in its decadal anniversary. And Thomas Hartmann had read the original German literature and reconstructed how it was that Ernst Stahl's ideas from 100 years prior had basically been buried as a consequence of scientists not wanting to be teleological, something that particularly after World War II was very problematic for scientists to make teleological arguments. And therefore, the argument that all of these metabolites were simply just waste products held the day until another German speaker, namely Gottfried Frankl, resuscitated that idea, published his paper in Science, and then that's what spurred this beautiful proliferation of theory about the function of the metabolites in the ecological literature. So to summarize things to where we are right now, this is a field with a deep history and a surplus of recent theoretical work. It was a body of work that's created largely by ecologists and zoologists rather than by plant biologists, and these outsiders brought a Darwinian perspective to plant function. And if you want to read some more, there's an article that I wrote in Nature in the process of reviewing a couple of books, and a beautiful article by Michael Pollan called The Intelligent Plant in the New Yorker that was published in 2013. Good reads on those topics. Now, what I want to do is to consider the plant-herbivore interaction in some more general terms. If you look at all different types of interactions that occur, that are sort of general ecological interactions, it's very clear that the most abundant interaction on the planet is that between plants and their herbivores. Plants, after all, are at the base of the food chain, and they comprise, a third or quarter of the described species. And insects that eat plants are another good 2/3 of those species, and all the other organisms that are herbivores from other taxa. So about 2/3 of all the organisms described on the planet are involved in this interaction between plants and herbivores. The fact that there's just so many plants that have to be eaten in order to sustain all the other animals that are on the planet. And as a consequence of this, it's worth considering the basic rules of plant-prey interactions to understand how they may apply to this particular interaction of a plant-herbivore interaction. So there's a couple different ways of dealing with your predator. The two basic ways of dealing with your predator are that you can either escape or you can defend yourself. And you can escape in lots of different ways. One common way of escaping predators is to escape in sheer numbers. And lots of organisms just produce tons of babies at the same time, so you overwhelm the ability of the predators to be able to eat all your babies. And you know lots of examples of mass spawning, and trees do the same thing. They have a process called masting, when all the trees in a whole geographic area will produce seed at the same time, and then they won't produce any seed for 7 or some long period of time after that. And the whole idea is you're just overwhelming the ability of predators to eat all of your babies, so some of them escape. So that's numerical escape. But there's also habitat escape. Lots of plants find particular habitats to escape their predators, just the way animals find habitats to escape their predators. And then there's escape behavior. Well, plants being rooted don't escape, run away too well, but there are other examples where plants can escape behaviorally. In addition to predator avoidance, there's also predator deceit, which is simply just to convince your predator that you're something that you don't want the tasty morsel that you really are. And you can do that through crypsis and camouflage, and you can do that through various types of mimicry, Batesian mimicry being one of the most common ones. Now, if you just think about animals for a moment, and you think about crypsis, here you have an example of a moth that is hiding on a tree and looking very much like the tree bark or the lichen that is on that tree bark, and therefore disguising itself. Plants do this all the time. It's just that a lot of times the animals that are hunting them are not using the visual dimension to see it, so we don't see it. But in the cases where visual dimensions are important, you can very easily see that cryptic behavior in plants. So for example, here is a weed on the far left-hand side, barnyard grass, which normally infests rice paddies, and when it infests rice paddies, it looks an awful lot like rice. So the barnyard grass outside of rice paddies is right here, and the barnyard grass that is in rice paddies is right there, and the rice, of course, is the far one on the left. And here, because human beings are the weeders, the barnyard grass has become cryptic, it looks like the rice, and is hard for human beings to see. This comes from a paper by Fred Gould, which really stands on the shoulders of research done by Spencer Barrett, who's also shown exactly the same thing happens with seeds. In the case of lentil fields, where a particular vetch seed that you can see down here, that's the vetch outside of lentil fields. The vetch over here is inside lentil fields, and the mechanical harvesters basically can't distinguish them. So this is just an example of the sort of selection pressures that plants respond to that are really no different from when animal prey are predated by other animals. The same thing happens with Batesian mimicry, and aposematic mimicry in general. We are all familiar with animals that are very poisonous, that stink or smell or have a chemical defense, and they advertise the fact that they have this chemical bomb in them with bright coloration, and that's all done through the visual modality. So skunks, for example, are black and white, and they raise their tails right before they squirt, and they let everyone know that they've got a really nasty chemical bomb in them. And plants do exactly the same thing, but they do it all through the olfactory dimension. And since we're not very olfactory creatures, we're not so familiar with the sort of Batesian mimicries that plants are doing. Now, so that's all the types of escape responses, but there's also the defense responses that plants have. And they're physical defenses, like the thorns and the spines that plants produce, and nutrient exclusion is another great way to defend yourself chemically. Simply just not have the elements in there that are essential for the digestive process. And since plant bodies are so different from animal bodies, for example, they lack sodium for the nervous system that animals need, or iron that animals need for their hemoglobin. By excluding sodium and iron, they can be quite poor nutritious things for animals, and that can function as a defense. Plants, of course, make all sorts of chemicals, and I'll talk much more about chemical defenses in parts 2 and parts 3 of the talk. And they also develop beautiful defensive mutualisms by which they hire ants with a little bit of sugar to stay on the plant and use the ants to protect themselves against herbivores that would come browse on them. The one part of defense that I want to describe in a little more detail is a process called indirect defense that is particularly special for plants because they use it so frequently, and it has to do with shaping the ecologic context in which defense occurs in plants. Now, it has been noted by ecologists for a long time that it's rather paradoxical that the world is green. The fact that the world is green most of the time means that herbivores are largely under control, either by predators or by the plants themselves. But there are lots of times when the world is not green, and herbivore populations grow really large and consume all the plants there, and all you have to do is read the Bible and you learn about those locust plagues, or you simply just go to a place where you're getting an outbreak of an herbivore, such as this gypsy moth outbreak that's occurring in Pennsylvania and completely denuded all the oak trees on these hills here, and when you go in these forests in the middle of the summer, all you hear is raining caterpillar poop because they're just turning all the leaves into caterpillar poop. And so there are plenty of times when the herbivore populations go out of control. And it was argued early in the 60s in a classical paper by Harston Smith and Soblakin that in general, the reason why herbivores are mostly under control and mostly the world is green is because predators and parasitoids are controlling herbivores. Well, that idea of top-down control was brought in by Peter Price in a seminal paper where he had identified and categorized the concept of indirect defense. So plants can directly defend themselves by producing chemicals that are noxious for the herbivores, and those are called direct defenses. But then they can also produce other types of information chemicals that provide information to the predators and parasitoids that are keeping the predators, the herbivores, in control, and those are called indirect defenses, and they allow the effect of the control of predators and parasitoids over the herbivore populations to exert themselves, and that's why the world is green most of the time. This is an idea, as I said, was first put forth by Peter Price and a number of colleagues in a very important paper in Annual Reviews in 1980. So what this means is that the plant-herbivore interaction, in comparison to the plant-pathogen interaction, is played out on an arena that is much larger than the plant itself. So if you look at study plant-pathogen interactions, it's primarily an interaction between the pathogen and the plant on a cellular basis. And remember, plants are in little wooden boxes, so that frequently the interaction has to do with this formation of a cell death response that you can see lighting up in this picture here. But in contrast, the plant-herbivore interaction involves a much larger spatial scale, involving predators that move around in a community and traits that from the plant that attract those predators that move around in that larger community context. Now, what I've been trying to argue for this talk is that plants solve their ecological problems through an innovative use of chemistry. And that plants produce lots of different types of chemicals, and that many of those chemicals are structurally just not understood, and most of them actually are not functionally understood. And what's really exciting about being a plant biologist today is that there's a quiet revolution in mass spectrometry that's occurring really as a consequence of Moore's law. The fact that computers are getting faster and faster means that mass spectrometers can scan faster and faster to collect data on chromatographic systems that are also separating molecules faster and faster, which means that of that very large, exuberant output that plants produce, we can begin to get mass spectral signatures on many of those molecules. We can begin to understand the metabolomes of plants for the first time. Now, the hard part is how to understand what plants are doing with all these wonderful chemicals. One thing is you can know what they are. The next question is, what do they do? Well, the way to think about how to understand that is that you have to start thinking like a plant. Now, in order to think like a plant, you've got to be able to ask the plant what it's doing with those chemicals. And the best way to ask the plant is first do something with yourself, and that is to think like a plant, to phytomorphize yourself. And what I mean by phytomorphization is that it's not the same process of anthropomorphization. In other words, taking human attributes and putting them into plants. What you want to do is to recognize that plants are different, that they have all those different things, as we mentioned earlier, all these different meristems and the fact that they're photosynthetic and like that, and begin to conceptualize the world as a plant would do it. Now, here's a good example which doesn't do it at all. The Secret Life of Plants is a good example of a book that anthropomorphized plants and provided almost no insights that were relevant to what plants actually do. And here's a good example of a book that does a beautiful job of phytomorphizing themselves. And David Attenborough's beautiful, stunning, slow-motion cinematography of trying to capture the time dimension on which plants are actually growing and behaving is a beautiful example of what you have to do as a biologist to sort of get into the mind of a plant. Now, you can ask the plant in the genomics era in a very sophisticated way. You can give the plant a question, an ecological question, and say, how are you going to deal with this? And then you simply query the various aspects that a plant responds in. It can respond in the way it transcribes its genes, translates them into proteins, makes metabolites, and changes its phenotype. And we have all sorts of beautiful ways of doing that, from very sophisticated chip technology to RNA-seq for the transcriptional responses, for the protein responses. We have many different types of gels. We have lots of different hyphenated mass spec procedures to be able to characterize these incredibly complex metabolomes, and we're getting better at phenotyping them, although that is in large part the slow part of this process, because we're still having a hard time phytomorphizing ourselves and understanding what plants really are doing. Now, if we want to understand the role that plant genes function in nature, we have to develop an evolutionary perspective on gene function. Understanding gene function is really the main question that biologists strive to answer in this particular era. But most of our understanding of gene function comes from protein expression studies, studies of which we understand how a particular protein functions usually as an enzyme, and that generates most of the gene annotations that we have in our libraries. But if we want to understand what actually maintains a gene in a genome and keeps it from being pseudogenized and lost through evolutionary time, we have to understand how the gene influences Darwinian fitness. And Darwinian fitness, of course, is the ability to produce successful grandchildren, and being able to produce successful grandchildren is something you have to study in the field, out in the environment in which the organism actually evolved. That's where real gene function can be understood and manipulated. And that is really the basis for the institute that we founded in Jena. What we wanted to do is to ask plants how they deal with the herbivores, and it was very clear that we had to take 4 disciplines that were normally separated in universities and different departments and put them together in the institute so that the scientists working in that institute could utilize all those different skills to be able to ask this question about how plants deal with their herbivores. And that is really the founding principle for this institute in Jena that I've been involved in. Here are the founding directors that helped form this institute in 1996. We were fortunate enough to locate it in exactly the same town and be associated with exactly the same university that Ernst Stahl was associated with when he did his seminal work 100 years earlier. Here's a picture of our institute in Jena. We first focused on how plants deal with herbivore attack. When the population geneticist Tom Mitchell-Olds decided to return, we had the opportunity, return to the US, we had the opportunity to broaden our perspective and look at how herbivores deal with plants, and brought in David Heckel to form an entomology department. So the 4 of us there were tending the goal. And then a few years later, we had the opportunity to bring in Bill Hansen and form a new department of neuroethology. Bill Hansen not only brought a different standard of sartorial activity or behavior to the department, but he also brought the skills and perspectives of neurobiology to the plant-insect interaction. And that I want to put into the context of the theory of plant-herbivore interactions that were developed post Gottfried Frankel's paper. One of the most important theories that came out was the apparency theory of Paul Feeney that he published in 1976. Now, Paul made a very insightful analysis that was based on fundamental evolutionary principles about the shaping of the type of molecules that plants have to use to defend themselves. And it is all based on the apparency of the plant. Trees, because they live hundreds of years, are clearly much more apparent to herbivores than an annual plant that only lives 1 year. Now, since insects frequently have short generation times, they have an ability to evolve resistance and counter a plant's defenses much more rapidly than a tree can. And that means that a tree has to come up with fundamentally different chemistry, a type of chemistry that is not easy to short circuit by a co-evolved insect. And it's going to have fundamental different properties, and that was what his basic prediction was, that the apparency of the plant would determine the type of chemistry that it had. But it had a problem. How do you actually know whether or not a tree is more apparent than an annual plant? And as Laurel Fox presented in a paper in American Zoologist in 1981, that there were basic operational difficulties with actually testing and falsifying this idea of apparency. Well, when we had the opportunity to bring in a fifth director and brought Bill Hansen in, we had the opportunity to see a plant through the nervous system of an insect and see how an insect actually perceives a plant and understand what makes a plant more apparent or less apparent to the insect through the insect's nervous system. After all, insects are very different from us, and we can't judge what an insect perceives based on our own nervous system. Okay, so we brought in this fifth department. This is the current constellation of the directors of the Max Planck Institute for Chemical Ecology. All 5 departments have their own model systems they work with. And what I want to do for the remaining part is really talking just about the system that we have developed in our department, which is the Department of Molecular Ecology. In this department, we have developed Nicotiana attenuata as a model system, which we explore. And Nicotiana attenuata grows in the southwestern deserts of the United States, in the Great Basin Desert. It occurs there primarily after fires. It chases fires in ecologic time. It produces a seed that is dormant, is long-lived, and waits for the next fire to come along, actually responding to wood smoke factors that stimulate its germination. And as a consequence of this particular behavior, it's a plant that grows in the primordial agricultural niche. The slash and burn agriculture was the foundation of agriculture for human beings, and this plant already figured this out a long time ago, probably about 4.5 million years ago. And it just basically waits for the next fire to come along and then starts its germination, synchronizes it with that wonderful nitrogen-rich soil that occurs after fires. And the reason for studying this particular plant is because if we understand how Nicotiana attenuata has grown in this particular niche, we'll hopefully be able to make our agricultural plants significantly smarter, engineer them for the type of stresses and challenges that growing in this particular niche allows them, so that we can have agricultural plants that require fewer inputs and the like. Now, to study this plant, we've developed 3 major platforms: a natural history, a metabolomics, and a molecular biology platform that have been headed over the various times by these wonderful people depicted below. The basic experimental approach that we take is that we start with a field observation, usually asking the plant about the particular ecologic circumstance. Then we try to get to the genetic basis of whatever the plant's response is, and then we try to transform the genes that are involved in that particular response, and having plants that are disabled genetically in that response, we take them back out to the field, do releases on them to test the hypothesis about their function. Now, there are basically 3 unique things about our department. The first thing is the training mission. What we try to do is to take early stage scientists, and instead of having them spend an entire degree learning all the particular techniques of metabolomics or molecular biology or natural history that they would normally in a university do in different departments, we fuse them together in one element here so that we can train a new generation of what we call genome-enabled field biologists. And what we mean by genome-enabled field biologists is very simple. What we want is somebody who asks organismic level questions about plants and their traits, but have, just like the 18th century biologists like Ernst Stahl was doing, and Alexander von Humboldt and Charles Darwin, but these students have a mass spectrometer in one pocket and a microarray in the other pocket, and have all the tools of molecular biology to ask those questions. So an essential part of what we do is this field station that we have that's located at the end of an 8,000 kilometer corridor that goes from Jena, Germany, right onto the United States, and in a field station that is located in a nature preserve run by Brigham Young University, we do the releases of the plants that we transform in Germany to ask these ecological questions. And in this field station, you can see it if you fly to Las Vegas from Frankfurt and look out the airplane window on the right-hand side. There is a couple of field plots where we do the releases of the transformed plants, but occasionally we also release plants into native populations depending on the question and the type of regulatory environment that we're working in. That's all controlled by APHIS, and these releases are all done under strict government supervision. The organization that governs this is the APHIS organization in the United States. Here's a picture of the field plot, and here's a series of images of the abiotic environment of how, after a fire cycle, these plants come, germinate, and form these large agricultural-like populations. And that's the abiotic environment, but just as important, and perhaps even more important, is the biotic environment, namely all the organisms that interact with this particular plant in its natural habitat are such an important part of our experimental procedure. Why? Because these organisms know how to phenotype our plants better than we do. They are much more phytomorphized than we are. Why? Because they make a living doing it. For most of the plants that we transform in Germany, they have no discernible phenotype. They look exactly identical to us. But when we take them out into Utah and plant them out in those field plots, those organisms help us phenotype them because they're specialists at doing it. And as a consequence, we're able to then use nature as a laboratory to study gene function. We have many examples where particular insects have told us about natural variation, and not to mention insects that specialize on eating different parts, sometimes stems, some bacteria. We're going to be talking about the bacteria in part 3. We'll talk about the predators in part 2. We'll talk about herbivores more in part 2, and we'll talk about seed predators in part 3 as well. But I could tell you about many of the other stories, and our publication record is another place to look for some of these interactions. So this approach is not really new. This is an approach that I learned from my mentor at Cornell University, Tom Eisner, who was one of the fathers of chemical ecology, and he was consistently melding excellent natural history with the latest technologies to be able to move the field forward. And we think that natural history skills are really one of the most essential tools in our toolbox as scientists to understand how things work. And this gets back to this issue of the combination of mechanism and function, that if you understand how things work, you can manipulate them, and those manipulations allow you to understand how a particular gene or a particular trait is functioning at the organismic level. Now, what we also do in our group is we silence genes in plants, and then we take them out into the field. But how do we actually find what genes to silence? And the way we do that is we simply ask the plant a question, frequently with an elicitor or a particular type of interaction, and then we look at how the plant responds both at the transcriptomic level and at the metabolite level. And we use the time dimension of those responses, that ripple and change in metabolites as a caterpillar attacks a plant, that happens in the metabolome, or that ripple and change in transcripts that happen in the plant's transcriptome as the caterpillar is attacked. And then we meld those together, and this is all done bioinformatically, and really is the genius of 3 people who've been in the department, Emmanuel Gagarel and Yosin Golati and Bepeng Li, who figured out wonderful informatic techniques of being able to generate gene-metabolite associations to look at how the genes and metabolites associate in time series of elicitations, and then develop networks to be able to find genes that are sometimes unknown but are regulating unknown metabolites. And by silencing them, we're able to then compare metabolomes and work out the structures of the metabolites. Now, what I want to do is very briefly end on an interaction story between 2 departments in our institute, the Hansen department and my department, on how a plant attracts pollinators. This plant, Nicotiana attenuata, is typically a night-flowering plant that produces a compound called benzolacetone from its flowers, and it uses that compound to attract a major pollinator, the Manduca sexta sphingid moth. Now, we first had to figure out how to make a plant that wasn't going to produce that particular scent. Klaus Gase looked at the transcriptome of exactly the part of the plant which releases the scent, which is the outer limb of the flower, looked at all the different transcripts, found a series of genes that were likely involved in the biosynthesis of benzolacetone, even though that was not unknown, did an RNAi experiment, silenced it, and generated a completely scent-free flower. And this scent-free plant we took out to Utah and planted them out with video cameras and anthraectomized flowers. And this looked at the interaction of the moth with the with those particular plants. And what it appeared to us was that the moths were largely ignoring the flowers that were silenced in the benzolacetone production and only going to visit the flowers that were producing benzolacetone. The ones that were producing benzolacetone produced seed capsules from outcrossing, and the ones that weren't producing benzolacetone were apparently ignored, at least in the pollination interaction. Well, Danny looked a little closer and looked at moths actually interacting with these chalcone silenced plants. In other words, they were not making benzolacetone. And as you can see from this movie, the moth has a very long proboscis, but is having a very hard time getting the proboscis inside the flower. It's taking that proboscis and bumping around on the flower, but not able to actually get it into the flower and orient it correctly. And he teamed up with some folks in the Hansen department, in particular Alexander Haverkamp, and they designed a very unique experiment that involved the ability to be able to separate the moth's normal apparatus for sensing benzolacetone, which are the antenna. You can see the antenna there on the moth's head there, from the ability of the tongue to sense benzolacetone, which you can see sticking into an apparatus there, which at the end of it has a Y tube. The Y tube allows for the introduction of the scent of benzolacetone in here versus normal non-scented air. So the moth is given a choice between two scented air streams after it's stuck its tongue deep inside this chamber. And in the next video, you'll see the tongue of the moth coming into this chamber. And as you can see on the right-hand side is the chamber that's scented with benzolacetone, and the moth's tongue is scenting down, searching there, testing out the control, but going back to the benzolacetone one. And the other neurobiologists in the Hansen department identified the particular the chemosensory sensilla on the tip of that moth's tongue that was responsible for sensing benzolacetone. And what it means is that the moth, while it's using its antenna to sense benzolacetone to orient in a long-distance way, once it comes to a flower, it's using the tip of its tongue to taste its way into the flower and be able to insert its tongue deep inside the flower. So this is a great example of why this field requires such interdisciplinary interactions. It takes all sorts of skills, neurobiologists, chemists, plant molecular biologists, ecologists, working together to understand how things work. And it also shows just how important this process of plants making themselves more apparent to the insects or the other animals that they're interacting with are, that Paul Feeney's idea of apparency was fundamentally correct as being an important organizing principle. And the fact that the plants are making themselves more apparent by advertising for pollination services, the insect is using that apparency, the fact that the flower is producing benzolacetone only in the corolla, as a way to help guide in its long tube inside to where it can find the nectar, which is why it's interested in pollination in the first place. Okay, so chemical ecology has a long tradition of experimental manipulations. I told you about Stahl's 1888 experiments treating plants with hydrochloric acid to dissolve raphides. That's a beautiful example of experimental manipulating a defense trait to see whether or not it's defensive. Chemical ecologists have been synthesizing the molecules that are involved in these interactions and using the synthetic molecules to recapitulate the interaction and study how that goes. But synthesizing a molecule and trying to get the organism to release it in the same way that it would normally is very difficult to do, and frequently those manipulations are quite ham-fisted. The real advance in the last decade has been the ability to bring molecular biology into this field to be able to silence genes that are involved in the production of those very chemicals that are mediating those interactions, and then take those gene silence organisms back out to the field into the natural environment and understand in an unbiased way by simply asking the plant or asking the ecosystem, what is this interaction all about? So the point is that genetic manipulations are absolutely essential for studying ecologic interactions. And this mechanism function interaction is really essential if we're going to make progress in understanding just how complex the ecological world really is. So that's all I'm going to say for part one. Part two, I'm going to tell you much more about this particular herbivore and that particular plant and what the plant does after it's recognized that it is that herbivore that's attacking it. Thank you for your attention, and also it's time for me to acknowledge not only the funding sources that make this all possible. The Max Planck Society and its long-term patient funding is the only scientific organization that would have funded this program, and the other organizations here, the European Research Council, the Human Frontiers Science Program, the Global Research Laboratory Program in South Korea, are examples of funding agencies that offer the least bureaucratic approach toward funding curiosity-driven research that I know of. I also need to thank the Brigham Young University for the use of their Lytle Ranch Preserve, and behind every brilliant university are exceptional people, and I want to particularly thank Dr. Larry Sinclair, Heriberto Madrigal, and Ken Packard for allowing that interaction to work so beautifully. And for all of the beautiful photographs, videos, and movies and presentation to support, I want to thank these individuals that are listed here, because without their photographic and stylistic skills, this talk would have been practically incoherent. Thank you for your attention.